quick review that the cell membrane is made out of phospholipids, which you know about the polarity of and the hydrophobicity and philicity of. That it's the fluid mosaic model that tells us that the cell membrane is kind of flowy like a soap bubble, but that it also has a bunch of stuff floating in it, including cholesterol that helps it stay fluid, and also the 3 different kinds of membrane proteins we talked about yesterday, which were transport proteins, which allow things across, like these simple channel proteins, marker proteins, which identify the cell to other cells, which your immune system needs to key in on, among other things, and receptor proteins, which do all kinds of jobs that have a little nook in them, kind of like the active site of an enzyme. And the example I gave you was your sense of smell, where the molecules plug in and send nerve impulses to your brain, which you asked lots of good questions about. Then we talked about the nucleus, and you learned what DNA does, sort of. You learned about chromatin and chromosomes, and the nuclear envelope, and how it's got holes in it, in that double membrane, for RNA to get in and out and copy genes and take their instructions out to build proteins with. And the nucleolus, which is where ribosomes get built. Now let's talk about the critters that don't have a nucleus versus the ones that do. This is going to be a chart. Pro is a prefix that can mean before. U means true. Karyo means middle part. So basically, prokaryotes are pre-nucleus. Eukaryotes have the true nucleus. So no nucleus is the thing that kind of first distinguishes prokaryotes from eukaryotes. They also don't have any of the organelles that we're about to start talking about. They have DNA, they have a cell membrane, they have ribosomes. That's about where they get off the bus. All the other organelles we're about to talk about, and color, that have membranes around them, bacteria, prokaryotes don't have. Do you know that metric unit? Do you know your metric units? I bet you know the ones about 4 or 5 slots either direction from the decimal point. Yeah. Micrometer or micrometer. And the metric signal for that is this Greek letter. That's not a U or an M. It's kind of a combination. They call it mu, MU. And so the way you do one of those is you kind of do the tail first and you go like that. And that's a mu. Okay? And micro means 1/1,000,000th of whatever unit it comes before. So in this case, it's 1/1,000,000th, 1 to 10 millionths of a meter. So they're little. They were also first. They are the first living things we have evidence of existing on Earth. 3.7, and that stands for billion years ago. How do we know? That's your next question, right? How can we possibly know what was there? Well, it turns out a lot of these bacteria formed little colonies that fossilize pretty well. They're called stromatolites, and there is evidence, fossil evidence, that we've dated roughly to 3.7 billion years old. It's just bacteria in this group. And by contrast, eukaryotes, yes nucleus, yes membrane-bound organelles. Bigger. Okay, math brains, if a micrometer is a millionth of a meter, what's 1,000 micrometers? 1/1000th of a meter. And what is that? 1 millimeter. You know, like the smallest ticks on your metric ruler? There are cells that are that big, that are actually basically visible to the naked eye, because they're a millimeter in diameter, but only eukaryotes get that big. The human ovum, the human egg cell, is one that's that big. Evolved more recently. The first evidence we have of eukaryotic cells is 1.5 billion years ago. And everything but bacteria belongs to this group, including animals, plants, fungi, and protists. 4 of the 6 kingdoms of living things. The other 2 are bacteria. And just for metric knowledge, let's write down that micrometer is 1/1,000,000th of a meter. By the way, three zeros finer is a nanometer. If you've heard of nanometers, that's 1 billionth of a meter. So we did the very outside of the cell. We did the very middle. We talked about who has the middle part and who doesn't. And now we talk about everything in between. What do we call everything in between? Nucleus and the cell membrane. The individual structures are called organelles. That's good. As a whole, what do we call that whole mess together? I think you know it. You look like you know it. You know? Yeah. Okay. Cytoplasm. I knew that. To distinguish, though, cytoplasm is like everything. If you're just talking about the kind of goo that everything's floating in, that's the cytosol. So you noticed in the kind of complicated drawing I gave you of the fluid mosaic model, on the inside of the cell membrane, there were lots of little fibers. And that's what I want to talk about today, is what's called the cytoskeleton. It's a network of protein fibers running throughout the cell. There's proteins in it. And the cytoskeleton has lots of functions, most of them involving movement, both of things inside the cell. The cytoskeleton fibers, some of them help move things around inside, and also of the cell itself. A different kind helps the cell change shape and maybe even move, either by oozing around or actually get on the outside of the cell and propel it like a tail. All of those things involve different fibers of the cytoskeleton. There's actually 3 kinds of fibers. We're gonna know 2. Microtubules is number 1. The 2 functions you need to know about microtubules are, first of all, they're almost like a little internal rail system, which you'll see in the video I'm going to show you. It's like you just need a little motor protein to attach to a big cell part, and it'll just walk it right along one of these microtubules, which, by the way, are tube-shaped. They're hollow in the inside, slightly bigger than the other filaments. So one way they work is by moving things around inside the cell. The other thing they can do is bundle up together and form things on the outside of the cell, hair-like or whip-like projections called cilia and flagella that actually help cells swim. Cells that swim typically do it one of 2 ways. Either they have a bunch of cilia, little tiny short hair-like projections, all up and down their cell membrane sticking out, doing this to move the cell through the water, Or they got one big honking flagellum. That's the singular form of flagellum, that's in the back of the cell doing this. So, cilia, flagella. That's my interpretive dance. Huh? No. But can you tell me the one human cell that has a flagellum? Sam. Bingo. Sperm cells are the only flagellated, I think is the word. It's a weird word. Cell. The only human cell with a flagellum. Good. So that's microtubules. Oh, there's a picture. I forgot I had a picture. Yeah. So cilia, flagellum. That's better than my interpretive dance. Microfilaments. Thinner than microtubules. Not hollow. These are the ones that are all about the shape of the cell. These are the ones that are anchored on the inside to the proteins running throughout the cell membrane to help it hold its shape. While the phospholipids are flowing around everywhere, the proteins give it some structure. This is the framework that supports the cell. So support, but then also movement in a way, because all you got to do is shorten certain microtubules and lengthen others, and you can change the shape of that cell. An amoeba is another one of those large cells that can be up to 1 millimeter. An amoeba is just a single-celled organism. There's lots of different species of them, but the way they move is all microfilaments. They basically just kind of ooze their cell, change its shape, and kind of ooze it out, and then the rest follows, and that's how they move. And it's all about shortening and lengthening microfilaments, this kind of what is called amoeboid movement of kind of oozing from place to place. Questions about the cytoskeleton? Here, here's a diagram, obviously kind of cartoony, showing though that microtubules, that's the big one here. Here's a mitochondrion, right? Mitochondria are you all know. It's like the only thing you learned about cells. And you can see it's stuck to that mitochondrion, and it's moving that mitochondrion from one place to another. Same with this piece of the endoplasmic reticulum. Here's a kind of cool colorized real microscope photograph showing the different sizes of filaments. And let's look at one more. Where are we? 30. Yeah. One organelle, and then we'll quit. It's the one that starts getting made in the nucleolus, and it's the one that RNA is going to when it leaves the nucleus. It's actually made of a special kind of RNA called rRNA. And when DNA gets copied into RNA, which is called messenger RNA, the messenger RNA leaves through the nuclear pores and goes here, goes to the ribosomes. That's a kind of general shape-wise drawing. Here's a more structural drawing showing that it is really just a big, long coil of RNA bunched up into that particular shape. But this is where it happens. This is where the thing that you drew for me in the very first chapter, amino acids forming peptide bonds until they're long enough to become proteins. This is where that happens. Save the video. It's a very cool video. I'm going to save it for tomorrow. And let's color. I'm going to give you a page from my favorite coloring book, the biology coloring book. Israel does exist. So you will need your colored pencils today. We're just going to worry about the front side today. And we're going to need some instructions here to know what we're doing. So, first of all, this is not just color whatever you want, whatever color you want. The top part is a key. And so the first job is to color each of the top terms with a different color. If you run out of different colors, you are welcome to combine colors for any of these terms. But so, you're going to color cell membrane, the word, one color, and then you're going to color nucleus a different color. All right, so then you got to match them up with the right cell parts. Now, that sounds scary because you don't know your cell parts yet, but they are labeled. Notice the little letters after each term. Notice that there's letters pointing to the cell parts on the coloring book page. Yeah? Get it? Now, there's a couple wrinkles here. First wrinkle is that there's a couple that have stars instead of letters. The ones that have stars are asking you to color the white space first in the nucleus, which there's not much, but second in the cytoplasm. Highly recommend you save the cytoplasm for last and simply choose a very light color like yellow so that you don't have to color around all these cell parts. Just kind of give it a shade over the dark stuff you've already done, if that makes sense. And no, you can't just leave it white. Next, if something is labeled once and you can tell there's more of them, for example, the mitochondrion, which is letter G, you might want to write in that it's actually smudged out on the left-hand side. Letter G is the thing that looks like a bean with a maze inside it on the left-hand side of the cell. Label that letter G, but also notice that there are two more of them. Those should also get colored whatever color you pick. That left-hand side is a problem. Let's go along the left and I'll tell you what the things are. Starting with M, you can see the M, right? On the left of the cell. The next one is an L. Then there's an O. Then there is a J. So M, L, O, J. Then K. Then that little guy there under the K is an O again. And then G, you already labeled. G is the mitochondrion. And then the next one down you should be able to see is a P. So M, L, O, J, K, O again, G, and then P goes down the left-hand side. I think that's all the special instructions. Good?